Greetings, my name is AIMAN, representing Center for Neuroscience Research, Faculty of Medicine, UITM. Here presenting my abstract entitled Effect of TRANS-RESVERATROL on Retinal Oxidative Stress in rats with NMDA-Induced Retinal Excitotoxicity. Here are the outlines for my presentation today. So let's talk about glaucoma. Now, glaucoma is an ocular disease that has affected the lives of more than 76 million people worldwide. Currently, it is the leading cause for irreversible blindness in the world. It can be divided into two main subtypes, which are people with normal intraocular pressure or IOP and those with elevated IOP. However, these two subtypes are potentially to develop glaucoma due to excitotoxicity neuronal injury, which can be caused by excessive influx of calcium ion that are caused by excessive synaptic glutamate. In turn, this will lead to retinal ganglion cell or RGC death that will lead to visual loss and finally leads to glaucoma itself. So let's talk about the normal physiology condition. We can see that the ion move freely from presynaptic neuron through the synaptic cleft onto the postsynaptic neuron. But in case of excitotoxicity, the excessive amount of calcium is diffusing into the postsynaptic neuron, hence triggered many apoptotic cascades. One of the main hallmark characterization of excitotoxicity is oxidative stress or OS. This happens when reactive oxygen species or ROS production overwhelms the intrinsic antioxidant capacity. In turn, it will cause damage to cellular macromolecules such as DNA, proteins, and lipids. And in time, it will cause pro-apoptotic signaling leading to glaucomatous neurodegeneration. My compound of interest is TRANS-RESVERATROL or TR. It is a polyphenolic compound. Previously, it effectively caused significant and sustained IOP reduction through adenosine A1 receptor modulation in rat model of glaucoma. It also increased the ganglion cell layer thickness and reduced the retinal oxidative stress in rat with elevated IOP. So my proposed mechanism of action for TRANS-RESVERATROL is that it will stimulate the adenosine A1 receptor to counteract NMDA-induced excitotoxicity. This in turn will lower calcium excessive influx, and this will lead to lesser apoptotic cascade triggered, hence lower oxidative stress as well. Interestingly, TRANS-RESVERATROL also have a direct effect towards reducing oxidative stress. For my problem statement, previously TRANS-RESVERATROL has been shown to effectively reduce retinal oxidative stress and IOP in rats with steroid-induced ocular hypertension. Notably, both pressure-dependent and independent loss of RGCs in glaucoma is known to involve excitotoxicity, and adenosine A1 receptor stimulation is known to reduce the excitotoxic neuronal injury. Although TRANS-RESVERATROL is known to reduce IOP via stimulation of adenosine A1 receptor, it remains unclear it could provide neuroprotection by reducing retinal oxidative stress via adenosine A1 receptor stimulation against NMDA-induced excitotoxicity, independent of elevated IOP. For my general objective is to determine if TRANS-RESVERATROL provide neuroprotection by reducing retinal oxidative stress via adenosine A1 receptor stimulation in rats with NMDA-induced retinal damage. For my specific objective is that to determine if TRANS-RESVERATROL reduce retinal oxidative stress through adenosine A1 receptor stimulation in rats with NMDA-induced retinal damage by estimating retinal levels of GHS, SOD, and CAT, and to determine if TRANS-RESVERATROL protects retinal morphology via adenosine A1 receptor stimulation in rats with NMDA-induced retinal damage by examining the H&E stained retinal cells. As for my methodology, the rats were divided into 4 consecutive groups. Namely, Group 1 is PBS, Group 2 is NMDA 160 nanomole, Group 3 is TRANS-RESVERATROL 4 nanomole with NMDA 160 nanomole 24 hours later, Group 4 is TRANS-RESVERATROL of 4 nanomole plus DPCPX of 8 nanomole with NMDA 160 nanomole 24 hours later. Now, DPCPX is a selective adenosine A1 receptor antagonist. 7 days later, the rats were euthanized and samples collected for ELISA, which are COD, CAT, and GSH, and H&E staining as well. So here are my results. Let's go through one by one. So A represents retinal CAT. As we can see in the TR treated group here, it showed higher level of retinal CAT as compared to NMDA group, indicating higher levels of activity for the CAT antioxidant. In contrast, for the TR-DPCPS group, it showed lesser levels of CAT, indicating lesser activity of the CAT antioxidant. Similarly with B, for the retinal GSH, the TR-treated group showed higher levels of the GSH antioxidant. This indicates higher levels of GSH activity. In contrast, the TRDP-CPS group showed lesser levels for GSH, indicating lesser activity for the GSH antioxidant. Last but not least is C, for the retinal SOD. The TR-treated group showed higher levels of SOD compared to NMDA group, indicating that it have higher levels of the SOD antioxidant. In contrast, the TRDP-CPS showed lesser levels of retinal SOD, indicating lesser activity of the SOD antioxidants activity. This proved that DPCPS partially abolished the therapeutic effect of TR. In correlation with the ELISA results, I also prepared a hematoxylin and eosin representative staining for each of the group. As you can see, indicated by the red arrow is the GCL layer. This is where all the RGC resides. As you can see in A for the PBS group, there are more numbers of RGC. In contrast, B, which is the NMDA group, there are few numbers of RGC located there. In contrast with C, which is the TR-treated group, there are slightly more numbers of RGC as compared to B. Interestingly, as we can see in D, is that the number of RGC is slightly lesser as compared to C. This indicates DPCPS partially abolished therapeutic effect of TR. So back to my proposed mechanism of action for TRANS-RESVERATROL. By editing DPCPX, which is a selective adenosine A1 receptor antagonist, will partially abolish the therapeutic effect of TRANS-RESVERATROL. So in conclusion, intravitreal TRANS-RESVERATROL protects against NMDA-induced retinal oxidative stress and changes in retinal morphology in rats. This effect of TRANS-RESVERATROL is likely to involve stimulation of adenosine A1 receptor. Further studies to determine the precise mechanism of action are warranted. I would like to thank Center for Neuroscience Research, LACHU, IMB, and the Ministry of Higher Education for giving me the chance to do this. That is all from me. Thank you. Any question from the floor?